Hello, everyone, and welcome to the Mindscape podcast. I'm your host, Sean Carroll. Cancer is one of the most terrible things we have to deal with in human life. It's a potentially fatal disease, of course. You know, we're all going to die someday. That's something that maybe we can make our peace with. But unlike many other diseases, cancer seems arbitrary in ways that are hard to pin down. It can happen to anyone. It can happen at any stage of your life. Young people can get it as well as old people. When you're reach a certain stage of your life, like I have, not only do you have to worry about checking for it yourself, but you know people who've had cancer and even who have died because of it. So it's very natural that as a species, we put a lot of effort into figuring out what is going on, how to stop this. It turns out to be really, really difficult, as maybe you know. Today's conversation is going to be with one of the world's leaders in the field of fighting cancer, James Allison, who won the Nobel Prize a few years ago in Physiology and Medicine for one of the various techniques that we can use to attack cancer once it starts. The idea being, rather than going in and just zapping the cells with radiation or chemicals or whatever, that we can use immunotherapy. In other words, we learn how to cajole the body's own existing immune system to fight the cancer tumors. And if this can work, it works in some cases, doesn't work in others yet. This is what we're studying, and we'll talk about it in the episode. But it's, not just a more kind of organic, natural way to fight the disease, but then the person who has had the therapy has that extra layer of protection against getting cancer going forward. You haven't just killed that tumor or most of that tumor. You've built up the body's defense systems. So, of course, that is, for real-world purposes, a fascinating and important development. For science purposes, of course, it's also fascinating because, man, the body, very, very complicated, very, very complex networks of reactions and cells and proteins and molecules and all that going on. We learn a lot about it because we are motivated for reasons of making people healthier, but what we learn is also equally fascinating. So we'll talk about how we got here, where we're going, and how I think the impression I get as an interested outsider is real progress is being made on one of the trickiest problems out there. So let's go. James Allison, welcome to the Mindscape podcast.
Thank you. Glad to be here.
I guess this is a big topic, right? I mean, you do research on cancer therapies and things like that. Let's start at the very lowest level here. We've all heard of cancer. It's bad. Something about cells dividing and going crazy. How do you think about what cancer is, broadly speaking?
I think it's a plague of a sort that unfortunately involves your own body going awry in some way, and a lot of I don't know. There's used to be a lot of stigma associated with it. Maybe there still is in some cases, but it's an unfortunate thing that happens to us. If we have a lot of cells that comprise our body. If they aren't controlled.
Yeah.
It's amazing to me that it works as well as it does, that everything stays under control. But unfortunately, it hits people. It hits a lot of people, way too many. It's hit way too many in my family. And
I guess the question I have is it the same phenomenon when we talk about different kinds of cancer? I mean, there's obviously a wide variety. I know some therapies work better than others. Is it accurate to call it one thing?
No, it's not. Well, it depends. I mean, the common thing is cells growing when they ought not to and where they ought not to. I mean, that's the common thing of all of them. But no, they're very different. They're different tissues, first of all. That's the way it used to be classified. Solid, and muscle or whatever, solid tissue versus leukemias, in the blood. And then now it's lung versus, colon or bladder versus prostate or whatever. But then with the advent of genomic sequencing and everything, the tendency was to attribute them to the causation. First, crudely, obviously carcinogen induced,
Right.
obviously virus induced or something. Although viruses are a tiny fraction of it, it's not zero, but that's not the main thing. But the common thing is just mutation, and the functional classification according to causation can be useful, and for a while it was thought was the real key. If like RAS is a molecule that really is involved in a pathway that takes signals outside a cell, like wounding or something, and it tells the cell, "Oh, you better divide because we need to cover up that scrape or that fix that cut or whatever." But if something happens and that pathway gets locked on, so the doorbell's ringing all the time, the cells just keep dividing. And the idea was, well, if we could inhibit that pathway, then we cured cancer, right? So this was all the excitement around the early 2000s, 2010 to up to about 2015 was most of these enzymes that do these or things are tyrosine kinases or forms thereof. And so the idea is if you can just inhibit that enzyme, you can cure the cancer by attacking the cause. Turns out, good idea, way too simple because the pathways always have multiple steps. And you can fix one of them, but then if there's another mutation downstream, you're back to where you started and your drug does nothing. And so that's what the problem is, because tumors, as they progress, as the tumors become more and more strange, as they become unstable, and the genome actually becomes very unstable, and you start getting a lot of more mutations. And as soon as that happens, you have more drivers, they're called. And so you can take one out, it doesn't make any difference. You can kill 99% of the tumor cells, it doesn't make any difference because 1% or 0.1% will have another driver and they'll inevitably grow out.
That's interesting. Actually, I don't think I knew that the rate of mutation in the tumor gets much larger. So as the tumor is growing, you're not just fighting one kind of cell.
Yeah, that's not totally universal, but generally it's true that the tumors become, as they progress, become less and less stable. And so the idea of having a magic bullet that can attack it at its source is really was not unreasonable, and was impossible to even think of doing anything about it until we had genomic sequencing, which we do now. But now we know it's probably not going to be. It's certainly worth doing because it can prolong life. As an immunologist, tend to look at things differently because the tumor biologists who take the classical tumor biology view of a cancer would say, "Oh, well, you got that mutation. Let's attack it, and we'll be done. So we should concentrate on what's going on there." On the other hand, there's lots of mutations. The mutations really occur pretty much randomly, and it's when they hit. So they've got to hit generally a couple of genes before you've got problems. A couple of very types of genes that are very specific, but the other ones, the cancer biologists say, "Oh, well, those are irrelevant. They're passengers, or they're not important because they're not drivers." The immune system, on the other hand, trained as an immunologist, your immune system is just, its purpose is to find things that shouldn't be there.
Exactly.
It doesn't care what they do. That shouldn't be on my cells. That cell may have a virus in it. I'm going to kill it. And so then you look at it that way, and those mutations, which to the cancer biologists are not important because they're not the drivers.
Yeah, okay.
They're equally important to the immune system. The immune system doesn't know the difference. It just knows there's something different here. We better get rid of that guy.
Well, you already mentioned that it's a little bit surprising to you that the body lasts as long as it does and works as well as it does. I mean, I see that, and I also see someone else saying it's kind of amazing to me that bodies haven't learned to fight cancer better since it's all over the place.
Yeah, well, evolutionarily, it's all evolution, and unless it takes you out before you reproduce, evolution doesn't care. If you get cancer when you're 50, evolution doesn't care.
Right.
It doesn't
Yeah, tell
matter
me
to
about
the
it. Right.
species. You know?
But some animal species are pretty good at avoiding
Yeah.
cancer. It does seem to be possible. Do we understand that?
No, I wish we did. No, I think that maybe they have a really good immune system. I doubt it. I doubt if it's that
Isn't there
simple.
some,
But
this is very vague to me, isn't there some paradox that larger animals you might expect to get cancer more easily because there's more cells, but in fact they get it more rarely?
yeah, I mean, that's true. I guess elephants don't, as far as I know, elephants don't get cancer very often. Uh, but then again, maybe they're not exposed to carcinogens like we are.
That's true. But mice, happily, we're able to give cancer to because
Yeah,
that's where we do a lot of our
happily,
tests,
yeah,
right?
not so good for the mice, but
So, how much do we know about? I know this is not, you sort of just very nicely explained why this is not what you care most about, but how much do we know about how tumors start? And is it just a myriad of various different reasons, or is there some central understanding?
well, there's some central understanding. I think what it comes down to, if you look at all the data that exists, is that it's basically mutations that cause it, and if you get out in the sun a lot and you expose your skin to ultraviolet radiation, the chances are you're going to get mutations in your skin that can cause melanoma
Yeah.
because that's what the sunlight hits, and if you stay out of the sun, you're probably not going to get melanoma. Although plenty of people, same with lung cancer, you smoke, you're highly, a lot more likely to get, but you're not. That does, if you don't smoke it, I mean, you're not going to get lung cancer
No.
because as cells divide, there are mistakes.
And you're obviously going to tell us about immunotherapies,
Yeah.
but maybe put that in the context of other kinds of therapies. I mean, we've all
Okay.
heard of chemotherapy, radiation therapy, et cetera.
Well,
These are the most
let's
common
go back
things we do.
historically. Let's go back way to the start. There's evidence that the Greeks, knew about tumors and they cut them off.
Okay.
And so the very first cancer therapy was surgery.
Got it. Yes.
And that still is perhaps the most effective if you can get it at all. That's the problem, though, because by the time certain kind of cancers in particular, like melanoma, very often by the time you notice it, and then you're kind of big whale, it's already spread to other organs in your body. Prostate, same way. So, but if you can catch it early, surgery is pretty effective. The next therapy that came along was radiation. The Curies around the end of the 19th century, beginning of the 20th century. Madame Curie, in particular, developed radiotherapy, and that was quite useful and curative in some cases. Of course, it also caused cancer. If
Right.
people learned by its mutagenic effects, but the idea was you blast a cell and give it enough mutations that it can't live. The problem, of course, is that it also kills the normal tissues. So you got to be careful about that. And then, oddly enough, with the advent of mustard gases and things during World War I and chemical warfare agents that were developed leading into World War II, ultimately led to some pioneers in cancer therapy in the 50s, applying it to leukemias, childhood leukemias in particular. Mustard, I mean, just it's toxic gases that toxic chemicals that were used to mustard gas, for example. Those were the basis of the first chemotherapies. They kill dividing cells.
Why
Which
are they good at killing
they
cancer
cause,
cells? Just
well,
because they divide
they
fast?
cause a lot more mutations, as they screw up the DNA as it's dividing and make it so that the cell can't successfully divide without making it.
Okay.
Unfortunately, you could also put in a lot of mutations that'll make
Right.
you more likely to get a cancer down the road too. But the bottom line with that, with both of those, both radiotherapy and chemotherapy as given, unless you kill every last tumor cell with those techniques, with those approaches to it, the tumor's going to win because it'll just come back. And so you have to blast them so hard or with radiation or poison them so much that it makes you sick, your hair falls out. the lining of your gut comes out. You don't make new blood cells. Your immune system's blown away, and you're sick. And that's what I saw when I was growing up, and my mother and two of her brothers, and
Yeah.
ultimately my brother, and it's a devastating thing. Not just the cancer, but the consequences of the therapies.
And
And
do
so
tumors spread just because the cells in the tumor sort of get carried around by the blood system, or
it's a little more complicated than that. There are signals that tell them to stay where they are. Those could be lost, but they can also develop things on their surface that'll tell them, well, you really belong over here. And
okay?
so it'll leave. I mean, that's the way they get there is through the blood, but it's more complicated. There has to be changes that allow them to get into the blood and other changes that allow them to get out of the blood into the tissue that they're going into. So that's a whole other area, metastasis of study that's underway. A lot of
And my understanding is that you started as a chemist, an actual chemist, more than a sort of biologist or
yeah,
medical person.
yeah. Well, a biochemist. I got bored with chemistry and biochemistry quickly, and actually, then when I was, but again, I had this family history of cancer, which made me. I was interested. Ultimately, I was interested in biology. Started in biochemistry because that's just I don't know where I landed.
Hmm.
But then, as I learned about the immune system, T cells had just been discovered shortly before I was an undergraduate, and I um, I was really fascinated by the whole idea of the immune system, but it was mostly antibodies and B cells at the time. But then when T cells came along, I was lucky enough to have an immunology course as an undergraduate at University of Texas at Austin, and Bill Mandy, the professor, towards the end of he was a antibody guy. Towards the end of the semester, he talked about these new cells that have been discovered called T cells that percolate all through your body. It was known, they go all through your body. I mean, not only just going around in the blood and the lymph, but they actually go through your tissues
Oh,
and
okay.
screen your cells and see what's going on. The remotest corner of your body, is being surveilled, I guess is the proper verb, um, by by the immune system to make sure nothing's going awry. And that's one of the reasons we're able to keep it all together without going completely bananas. One of the reasons early on. First of all, most of the mechanisms for replicating are pretty damned accurate, but if they're accurate 99.9% of the time, that's not enough.
Not enough.
You're
Yeah.
going to get problems.
And T
Give it
cells
all the
are
cells.
they're a variety of white blood cells.
Yeah, yeah, a variety of them. Yeah, lymphocytes are one kind, but they're also macrophages. They're largely, I guess I would say, two families of them: lymphoid cells and myeloid cells. Myeloid cells are like macrophages and things like that engulf bacteria or infected cells or dying debris from dying cells and clean up wounds and all that stuff and help wounds repair. Um, and they have innate signals, they're called, that they can recognize a lot of viruses, a lot of bacterial, just because they have carbohydrates and things on their surface, that is classes of molecules are different than those found in our cells, mammalian cells. So they're sort of this, we call them the innate because everybody's got those and they can protect you against a lot of
Yeah.
organisms. But that's not enough either. And so late in evolution with development of actually not shortly after vertebrates and things. But anyway, evolutionarily, this other system came along, which we call the adaptive immune system, where you actually have receptors that are made by random recombination of DNA sequences. I don't want to go into the details, but basically, essentially, random processes that give you a random set of different receptors. It's been calculated that you can make all things, if you had full ability to make every receptor that can be made with the structures that are in there. And it's really a fascinating area of biology that
Mm.
in its own, but it's 10 to the 15th maybe 10 to the 17th power.
Yeah.
You only have about 10 to the 10th cells in your body, 10 to the 12th cells. So it's 1,000 times more cells than total cells, at least 1,000 times more total cells than you have in your body. So each of us really realize only a fraction of the possible diversity of different T cell receptors that could be made in your body and to the species, you know. So the population is protected probably, but not the individual necessarily.
Well, I was going to ask, do the T cells in our body develop new receptors? Do they learn on the job, or does the body make new T
They're
cells when
generated.
new?
No, they're generated essentially randomly.
Okay.
The cells come out of the bone marrow. For T cells, they come out of the bone marrow. They go to this organ called the thymus, which sits right above the heart. And there they start developing from this precursor stem cell into functional T cells by random rearrangements. And they're
Oh,
put together.
interesting.
And there's this fascinating testing system where there's a scaffold on cells that presents the antigens, which are little bits of protein that are only 8 to 10 amino acids, 8 to 12 maybe amino acids long, that are presented in the surface of this thing that looks like a hot dog bun. And it's got a peptide in the middle of it there's a peptide for virtually every protein that's made in your body that'll bind there and will be put on the surface of your cells. So the immune system knows a sample of everything that's going on in the cell, even if the thing normally wouldn't be on the cell surface. If it's
Okay.
a virus that's just infected a cell and the cell starts making bits of virus, making the parts of the virus that it needs to reproduce and go out and infect other cells. As they're making it, those proteins will be cut into little pieces, and pieces of them will be put on the surface. And if the right T cell comes by, it says, "Whoops, that shouldn't be there," and it'll kill it. It's
So
essentially
basically,
random.
so the cell is sort of doing its own annual checkup at all times.
Exactly. But you got to get rid of a lot of those are going to be harmful. They may be harmful. And so they got to, the tumor, the thymus not only educates the cells as to which ones can be useful, but gets rid of the ones which will hurt you, hopefully. If they don't, you end up with diabetes, or you end up with, different autoimmune syndromes and stuff. That's where
So, sorry, these are T cells that can be dangerous if they're not in the right kind?
Yeah, if they're autoreactive, if they react
Right.
with cell proteins. Or sometimes if a virus comes in and tricks the immune system into thinking something's foreign, when it's closely enough to a self thing, occasionally you'll get a spillover, can be damaged, like from measles and stuff, down the road. But anyway, basically they're pretty damn good, though.
Yeah. This is where my simple physics brain rebels at the complexity of all the networks inside the human body. It's kind of an amazing edifice.
Yeah, how many stars are there? 10 to the
Well, there's a lot of stars, but stars are pretty similar to each other. There's 10 to the 22 stars out there, but they're not that different.
No.
There's no lock and key in there.
No.
I guess that's my question. Your talk about receptors reminds me of people who are trying to study smell and how we're sensitive to different kinds of molecules. Is it a similar kind of thing going on?
Yeah, in a way, except that there, what you have is one kind of receptor for each kind of bit of a smell. And it's the sum of all those that tells you what the overall smell is. But each one only detects one thing, and the brain integrates it all.
Yeah.
Whereas here, you've got all these different ones that are flowing all around. All you need to do is trigger one.
And so the job of a T cell is to understand what the normal healthy cells are like and target anything that is not that.
Exactly. Not self-recognition is what it's called. Very philosophically, how do you recognize not self?
But they do a pretty good job, like you said, and yet we still get cancer. So there's some reason why, I guess in principle, they can attack cancers, but they don't do as well as they could.
Yeah, that's first of all because the cancer cells are not necessarily all that different early on, especially. although they get weirder and weirder with time, and often with a lot more mutations. But they also have ways of protecting themselves, because cells don't like to be killed either. Um, for example, in tumor cells, there's a process called apoptosis, and there are mechanisms that guard cells built into the cell, or mechanisms for detecting mutations. And if there's too many, the cell tries to commit suicide. It's told to kill yourself because you're going to may cause cancer. At least that's the thought. But there are these suppressor genes which do that. So really, in order to get cancer, you've got to not only get an activating gene, which will tell the cell it ought to be a cancer, but you got to get rid of those suppressor genes,
Right.
which would shut that down. So it's genetically, it's complicated too, because you really have to have both in order to
I
get
mean,
it.
I,
So
I
that's why
guess
some
this.
people with retinoblastoma gene, for example, if you have two copies of that, kids get tumors of the eyes when they're about 2 years old. It's a devastating disease. But in other kinds of cancer, you don't get them in your germline, but you can get them in your somatic cells. And if you lose the RB genes, that makes you that makes that cell a lot more likely to get cancer. To
That helps
support.
because I did have the question, do tumors or do cancer cells defend themselves? They don't pass on their genes in some sense.
Right.
But I guess the answer is, but they're versions of or made of ordinary cells, which do have defense mechanisms.
Yeah, yeah. And they also, one of the things that we found recently that's even more interesting to me is that the immune system, every now and then, these macrophages who play a role in cleaning up after wounds and wound healing and replacement, they'll protect the tumor too. They think the
Oh, okay.
tumor is a wound. And so your own immune system can turn around. And we're finding that that's one of the reasons that happens big time in pancreatic cancer and in um, glioblastoma, which are tumors that are very lethal. And we're still, we got the, it's not that we got the T cell issues solved with those, but what we know is there are myeloid cells there that are trying to stop the T cells from killing the tissue because they're just doing that's their normal function, is protect tissue and help it heal. They just
Right.
happen to, they get in a weird situation where they are treating the tumor like a wound and protecting it from the immune system. So that's one of the things that we're working on now, is trying to find out some way to override that or change the myeloid cells where they'll help. They can also help the T cells. It just depends on what they're doing. It's a whole new area of biology that single cell RNA techniques and things are making us realize that. And you talk about T cells, there used to be B cells and T cells. Now there are T cells specifically. First there were 2 kinds, then there were 5 kinds. Now there are about 7 kinds. The truth is that those kinds are just constructs that we make up.
Yep.
In biology, it's a continuity. It's just a continual spread. In myeloid cells, it's really pronounced. You can see all these different cells. They differ by 2 or 3 genes that are being expressed. And you can just shift the population one way or another. And that's what we're trying to figure out. And then shift them from this protective overall effect to the helping, thing to help the immune system rather than hinder it. So
One impression I get from reading about these or talking to people is that everything is about switches, turning things on and off. Like once you realize that every cell has the same DNA in it, I guess it makes sense because they do very different things, but it's all a matter of which parts of them are playing a role at this particular moment.
which genes are turned on and how much, and it's the relative amounts of different genes in some cases. The complexity is amazing, and so our brains cut and box things, but nature doesn't.
How much, it sounds like with the receptor stuff on the wall of the T cell, that's right down to the level of atomic and molecular structure, right? Is it very different how we're studying that now than when we were in like the 1980s or whenever it was? Like, has the technology changed things?
Well, yeah. I mean, in the 70s, nobody knew what the T cell receptor was. I mean, that was my first thing when I got interested in immunology was what is the antigen receptor? What is it that the T cell uses? And nobody knew what it was. And so I put on my biochemist hat and said, well, what should such a molecule look like? And worked it out and published that for the first time in 82. It's going to look like this. And I had data and stuff. And then the genes were cloned a couple of years later and confirmed that's what it was, an alpha chain and a beta chain, two chains of protein, both of which have constant regions and then variable regions. The outside that gives you the combining site. But how is it presented? And so there was this, I won't go into the details too much, but that's another complicated thing, how the molecule, that hot dog, it doesn't, every wiener won't fit into that bun. Everybody's bun's a little bit different. It holds a different kind of wiener. And so, if you've got the right, bun for the peptide, you can present it. If you don't, you won't. So, that's another, if you can't present a peptide, even though it's there and it's from a cancer, then the T cells aren't going to see it. So
we don't mind a little bit of details here. You're allowed, like, if you really want to go into details, we'll go with you a little.
Okay,
way,
well, I won't go anymore. But it's a complicated thing. But we didn't know this stuff until the 80s and the 90s, and now we're beginning to understand in some detail. And now with a lot of the work in terms of really understanding the biology of it, some of this the new work from the Nobel Prize this year, David Baker, for example, in understanding how sequence informs shapes and how shapes influence sequence. All this we're beginning to understand at the molecular level, how these interactions occur and how to manipulate them a little bit better in ways we couldn't even think about 10 years ago, much less in the 80s.
I do love the idea that even before you really had any direct evidence of what it looked like, you could sit back and think, well, what should it look like to do the job that it does? In some sense, that's what people did for DNA too.
Yeah, I'm one of these old school guys that thinks hypotheses are useful.
Yeah.
Now it's get a whole bunch of data and try to pull something out of it,
Well, are we at the level now where we take pictures of not just T cells, but the actual receptors that are on them?
Yeah, pretty much. Pretty much.
Those are the things that have 10 to the 17 different possibilities.
That's really helping with vaccine design.
Right.
And stuff now. So we're on the verge of being able to, in some cases, develop prophylactic vaccines, I think. Certainly for HPV, we're there. We can prevent HPV-induced cervical cancer and head and neck cancer. By the HPV vaccine, if it was people would just use it. More importantly, in the clinical realm, are therapeutic vaccines now, and we're beginning to understand enough about the antigens that are induced by the mutations, where we can begin to come up. This was something that was greatly informed by our recent epidemic, with the virus, with the vaccine strategies that were developed then, where you've got basically a cassette approach now, where we know how to really put a gene in the middle of this cassette. I mean, I think we're going to come up. There's going to be eventually a small set of adapters, you can use. It'll have all the signals that you need to properly activate the innate immune system and all that. But you put your specific thing in the middle of it, and we can generate a vaccine in weeks.
Pretty good. I mean, is this part of the sort of CRISPR revolution of gene editing?
That's part of it.
Yeah.
That's part of it, being able to get the genes out. It's more useful right now in testing as to whether you're right by actually doing fine structure and seeing if you can initiate or terminate an immune response by manipulating things. But yeah, it's that and just all the work that went in developing about the coronavirus vaccines, the COVID vaccines. But that sort of framework, it's not exactly the same thing, but it'd be something very similar, which now you can theoretically take a melanoma patient, sequence the genome, and with computer algorithms predict which of the mutations would actually elicit in your own body, because different people's, it's called MHC type, the genes that you have that influence what you present. If you know that, and you know the sequence of the different peptides, you can predict which ones will be presented, and then you can design the proper vaccine to snap it into that thing and start vaccinating people. So that's coming along. I think that preventive vaccines, pretty soon we're going to be able to maybe protect people that have what's called what is it, Lynch syndrome. They get a lot of polyps,
Oh
that
yeah.
eventually go on and develop colon cancer. It's definitely a predisposition you can predict. But there are mutations, common mutations associated with that I think if you immunized early enough, you could protect those people from ever developing the disease by immunizing them later. Maybe the same will happen with BRCA. BRCA mutations are associated with in women with breast cancer and ovarian cancer, and men also with certain kinds of cancer. But it may be possible to prepare vaccines there. But most of the times, the mutations are so individually specific to you, that there's no way you're going to have a vaccine you can prophylactically
Right.
treat the population for most cancers. You're going to have to come up with something that's really powerful and allows you to jumpstart a response as soon as you detect it. And I think we're getting close. We're not there yet, but
But I was actually going to ask about the role of simulations. I mean, as a in physics and astronomy, of course, that's what we do all the time. We simulate things, we test them against the data. I've always had the impression that in biology, the state of the art was that biological things are too complicated to do that and too specific and too individual. So we have to actually test the pharmaceutical in a living thing rather than just putting it on a computer.
yeah, I mean we're still there. Although it's better now, we can guess. We can
Okay.
make educated guesses now. We can't get exactly there, but
So we have this basic security force in our body of the T cells roaming around looking for interlopers. Um, you mentioned a little bit about this already, but why is it that they aren't better at attacking cancer, and how can we make them better? That seems to be the project, right?
yeah. Well, again, it's because one of them is big, and it's largely they're individual. So your tumor has mutations that nobody else's has. So you got to tailor it to the individual. And secondly, there are ways that the tumor can lose. For example, the peptide has to be from the containing the mutation has to get on the cell surface by these MHC molecules. And if you lose expression of MHC molecules, then they're invisible to the immune system. The tumor will still be there, still making the mutation will still be being made, but the immune system can't see it because it doesn't get to the surface of the cell. There are ways around that we've got to have them, but that's what happens and has been shown in melanoma, for example. Patients
Okay.
um, just quit making the molecule that carries it. But there are other ways around that we're beginning to come up with. But that's one easy mechanism. Another one is that one of the main ways that tumors kill tumor cells is by making gamma interferon, which is a cytokine that, I mean, you've heard of, but one of its activities, besides helping protect you against viruses, it'll causes tumor cells to quit dividing and can kill many of them. But if you lose anything along the receptor downstream of the gamma interferon, the thing that detects gamma interferon and tells the cell it's around, if you mutate anything in that pathway, then that doesn't work anymore either. And
Hmm.
so the T cells can make all the gamma interferon they want, and the tumor you know, won't respond to it. So there's that too.
So what is our goal as immunotherapists? Are we trying to teach the T cells to ignore some of these
well, no,
problems?
first it's just to get the T cells.
Yeah.
And then secondly, in importance, is do something about the myeloid cells, which we're not there yet. we're getting close on the T cells. The myeloid cells, we're only beginning there. But ultimately, it's how do we deal with these things that comes down. So um, it's not that you can get the T cells to do any better, but you can come up with ways of making them work at a distance. It's kind of hard to explain, but a different type of T cell that'll make more soluble factors, maybe see the antigen on a myeloid cell, even though the tumors can't present it. If these myeloid cells, which will be gobbling up, a tumor cell dies, they'll pick up pieces of it, put it on their surface. If they have the antigen and a T cell sees it there, and they can make enough of these gamma interferons or other things, then they can kill the tumor cells at a distance, even though they don't interact physically directly with the tumor cell anymore. I mean, we can show that happens in animal models. At least I'm pretty sure it happens in people as well. But
Okay, I mean, I read a little bit about the T cells before talking to you, but I didn't read anything about myeloid cells. Maybe you should tell me something about that. It sounds like they're important.
yeah, that's the reason. I mean, I used to avoid the things like a plague because they're so complicated. There's so many different types, but what we found I'm speaking there not as just me, but the field began to realize is the tumors which don't respond well to T cell based therapies usually have really large populations of myeloid cells in them. So that raised the interest. And sure enough, you can show that the myeloid cells can inhibit T cells.
Hmm.
And we've found two molecules on myeloid cells that do that. And we've shown if you take them away, then we can make the T cells more effective. So that leads rise to some more strategies where you target the myeloid cells, but you're going to have to do both, particularly in things like pancreatic and glioblastoma.
What are the myeloid cells? What's their role when everything is going well?
Gobble up bacteria that come in, deal with if you've made antibodies and they're binding to viruses or bacteria and clumping them up to gobble up and get rid of the bugs that way. As I said, to help wounds heal, to
Okay, good.
make growth factors and stuff, just sort of general handymen. They're just sort of general handymen that do whatever fixer-uppers or you need, but they can get in the way.
And they have this spin-off effect that they can basically communicate the existence of a tumor to the T cells?
If you treat them right, yeah.
Yeah.
They can hide them from the tumor cell too. I mean, they can hide the tumor cell from the T cells too.
Right, if things are going badly. And
Yeah.
isn't there also, this is my very vague understanding popping up, but you can get in trouble if you make too many T cells.
Yeah. If you make too many T cells, you can definitely get sick, and especially if they react with normal cells or with something that makes a big soluble factors that affect. And that unfortunately is one of the main side effects of the therapies is cross reactivity with normal tissues and things. That I mean, there's no free lunch. You
Right.
start messing with those things. It's not that it always happens. I know of at least one marathoner who got melanoma and went through the whole course of therapy and never missed a race.
Oh, wow. Okay.
Other people get very sick, and sometimes there can be things which can be fatal. Thank God they're rare, and the clinicians now know from experience. Have just developed algorithms for recognizing when it's about to happen and heading it off, or at least reversing it early on while you still got time.
And by the therapy, I mean, it sounds like what we're doing is sort of trying to regulate the amount and the sensitivity of these different cells in our immune system, presumably by giving people drugs.
Drugs in the form of, right now, one of the popular things are antibodies. These are just proteins that we can make that are very specific. I mean, that's what we did with CTLA-4 in the 90s. There was this molecule, and it was funny. It looked like a molecule that was the gas pedal on T cells, but this molecule called CTLA-4, and we showed that it was actually not a gas pedal, it was a brake.
Ah.
And that the people that called it a gas pedal, what they did was they had T cells, they put them in culture, activate them, and then add an antibody, and they'd get more. And they said, okay, well, that's a gas pedal. That's what we did early in the 80s, showed that CD28 is another molecule. It's like the gas pedal. So that antigen receptor you can think of as the ignition switch. That's
Okay.
all it is. It tells a T cell. But a T cell has to get a second signal at the same time, which is like the gas pedal. And that's a molecule called CD28 that we showed in the late 80s, was the gas pedal. And if you don't push on that, activating through the antigen receptor doesn't do you any good at all. The T cells don't do anything. Tumors never have, on their solid tumors, I should say, don't have the structure that binds to that gas pedal. So it's complicated. So solid tumors are invisible to the immune system because they don't have that second signal. The only way they get them is to the tumor. And we worked this out in the early 90s. The tumor gets big enough or whatever causes inflammation. The innate immune system comes in. The innate immune system has those things they're called B7 for. They can be called Frank. It doesn't matter. I mean, it's just the name means nothing, but they bind to CD28, and that says go. So the only way that the immune system with solid tumors, and this is we published this in the early 90s, was that they grow until there's tumor cell death, and the innate immune system comes in and primes the T cells, and then you start generating T cells. But the thing is, you probably got 10 to the 10th, 10 to the 9th, different, nobody really knows for sure, but different T cells in your body with different receptors. And of course, that means you've only got a few hundred, maybe, of any given clone. And that's not enough to protect you against anything. You need hundreds of thousands. Yeah. And so they have to expand. And so that's what so the T cell receptor sees the ignition switch, and then but nothing happens really until you push on the CD28 molecule, the gas pedal. Then they take off. You got to generate 100,000. I mean, you've got hundreds of thousands to millions of cells to swarm through the body and look for things. I mean, that's what's so cool about it, because then you don't need to know where to cut or know where to shine the radiation or whatever. You let the T cells go find it, go find the micrometastases, the little clumps of tumor cell under your toenail or wherever. I mean, that's an exaggeration, obviously. But anyway, they go find the tumor cells wherever they are and take them out, or at least keep them in control where they don't grow anymore. But that process has to happen fast or the tumor wins. And so,
because tumors are going to grow very fast generally, yeah?
Right. And see, well, they not that fast. It takes years generally. It's at the end when they start damaging.
Oh, okay.
That's the problem. I mean, a lot of tumors can get quite large as long as they aren't in a place. I mean, you got a lot more degrees of freedom in a tumor that's in your abdomen than a tumor's in your brain, for example. Just the sheer pressure in the brain will kill you.
Right. Is it something where different kinds of cancer are still going to need different kinds of T cells to come to life?
Not grossly different. They're basically the same kind of T cell. So there's commonality, but it's those myeloid cells again that get in there because they can interfere in different ways. So we've got to work on those. But anyway, there's this other molecule called CTLA4, which we showed in the early 90s. Again, was a break. And so at the end of that expansion phase, that CTLA4 molecule starts coming up and stops the T cells from dividing. They've got to stop, or they'll kill you.
Yeah.
Because they just, they got to stop. I mean, it's not, they don't become a cancer. I mean, it's nothing like that, but they'll just grow normally and use up all your nutrients and nothing left for everybody else. So you got to stop that process. And that's what this molecule called CTLA4 does. And so the thing that we figured out in the early 90s was if you block that molecule, you can let the T cells keep going a little bit longer than they would normally, long enough to take out the tumor. And then you stop the therapy and everything comes back to normal. And so that works spectacularly well in some kinds of cancer. Just taking the brakes off for a while.
Right. I mean, I know that one of the most terrible things you can hear when you have cancer is that it has spread, right?
Yeah.
It's spread around the body. So it sounds like maybe this kind of therapy will be more amenable to even dealing with that.
Yeah. Melanoma kills you because it, ends up in the liver, or the bone, or the brain. Jimmy Carter had a melanoma in his brain. He
Hmm.
got immunotherapy, was cured. Lived to be 100 and whatever, 100, I guess
Oh,
he
okay.
was 100. He was cured about 7 years ago with immunotherapy of brain cancer.
So I guess that answers my next question. Like, how much is this in the clinic now? Is there a pill that you can take? Is this growing?
Yeah,
Is it being
no,
tested?
it's not. No, it's all over the world. There are millions of people, literally, that have been treated. In fact, in melanoma now, immunotherapy is pretty much the standard of care. It's the first
Good.
thing you'll get because it's so effective. The drug that we developed in the late 90s, ipilimumab, that was approved by the FDA in 2011, that's given all over the world. As I said, millions of people have been treated, and it cures overall by itself about 20% of people with metastatic melanoma. To put this in context, melanoma was one of the earliest targets for several reasons, but one of which is no drug had ever had any effect at all in melanoma, ever. And so when you go with immunotherapy, and also there were some indications that maybe it was immunogenic because it has a lot of mutations. But in any event, with our drug, 20% of people are cured by one or two injections of an antibody. The therapy, by the way, is you sit in a chair for about an hour, and they run there's a drip bottle with a drug in it, and it's infused in your blood, and you go home, and that's it.
And that's better than chemotherapy as we know it.
Yeah, and with luck, most patients there's some scratchiness, there's some diarrhea, there's some stuff, but usually nothing really bad. Occasionally there can be bad stuff. Even some patients get type 1 diabetes, which is an autoimmune condition. It may be that they already had it,
Yeah.
borderline, and didn't know it. It just makes it worse. But I mean, there's a downside. A lot of patients, but most of the time it's not. Anyway, it was 20%. But then after we started, our stuff started coming out. Tasco Hanjo in Japan, plus Arlene Sharp and Gordon Freeman at the Dana-Farber discovered this other checkpoint. CTLA-4 was the first checkpoint defined as a cell intrinsic, a molecule on the surface of a T cell itself that helps give a negative signal.
Okay.
So the T cell receptor obviously is a positive signal. It's the distance switch. CD28 is the gas pill. It's another positive signal. CTLA-4 says, stop all that stuff. It's time to quit. And so that's the one we chose to focus on. Was you can either make the other ones better. We chose, let's just take the brakes off. Let's disable the brakes. And it worked spectacularly well. As I said, in the phase 1 trial, which normally is just safety. You make sure you're not killing anybody and then titrate it up until they start getting really sick and then back off a little bit. And that's the way that cancer therapy used to be. So there you prove its safety. You find the maximum tolerated dose. How far can you go before you make people sick? And then you give that dose and you give it until all the tumor's gone. And if the tumor grows at all, it's a failure. All of that's out the window with immunotherapies. First of all, there is no maximum tolerated dose usually. People they may have adverse events, but, you go up and up and, you might it gets more frequent in the population, but people don't necessarily get sicker. It's not like there's a poisonous level of it. And in melanoma, in the first 14 patients, phase 1, there were 3 whose tumors completely went away. Which was just unheard of at the time, particularly in melanoma, because in 2011, when the phase 3 trial, that I was associated with, was unblinded and reported to the FDA. At that time, if you were diagnosed with metastatic melanoma, the median survival, 50% of people would be dead in 7 months. Fewer than 3% would be alive at 5 years. After that, it was minuscule. It's not to say that everybody always died, but it was much less than 1%
Yeah.
would survive. Now, with just this one drug, 20% are alive at 10 years plus.
Mm.
With no other therapy, just one round of therapy, you're done. When Tosca came along with this other molecule called PD-1, which works a slightly different way, but the overall picture's similar enough. It's another checkpoint that works differently. If you put them together, there was just a trial that was just reported about a month ago. There was over 1,000 people randomized, 10 different countries. I don't know how many different PIs. I mean, the gold standard of clinical trials, 10 years follow-up, 55% of the patients were still alive.
Wow.
So now we went from a cancer, which was almost uniformly fatal in less than 5 years, to we can cure more than 50% of the people with that disease.
That is amazing. But so my immediate reaction is that 20%, 50, 55% numbers are, on the one hand, super impressive. On the other hand, why not 100%? What do we got to do?
Exactly. And that's exactly what we're working on now, is how do we get that to 100?
Yeah.
And unfortunately, I don't want to get into that, but I'm not sure that the drug companies seem to be happy with 55% or so.
Yeah.
Anyway, it gets harder now, but that is the question, right? I mean, to me, that's why we have this thing we have this institute that's been founded in Anderson, and its whole goal is how do we make that better?
Right.
And the way you make that better, again, is by bringing the myeloid cells in there. What are the ways? What are the things that agree with it? Now we know, for example, there are a lot more of these things called checkpoints. Probably, I mean, there's a small number. It's probably a dozen, maybe. I don't know for sure, but that influence the immune system in various ways, that they regulate it. And some of them only pop up when you take one off. In one sense, it's whack-a-mole. You take one off and another one, because the immune system tries to regulate itself. I mean, the biology is just wonderful, just wonderfully complicated, and the way it all fits together to make sure that everything that can happen, there's a counterweight to it. Multiple ways are built in. But because it doesn't want to kill you. I mean, there are more ways of turning an immune response off than there are turning it on, simply because the consequences of having it work when it shouldn't are too devastating, particularly if it kills you when you're young, So it's all tuned to do that. But anyway, so one of the cardinal rules in pharma drug development is make sure something has a single agent activity before you combine it with something else. That paradigm is shot here because some of those molecules aren't even expressed until you give one of the other ones. And so we've got to get off of that, off of that sort of thing and understand it mechanistically. And so what we're doing is going into humans as soon as we can, as soon as we know it's safe, and giving combinations and getting biopsies and seeing what happened and what new molecules came out. Because now we can measure the advances that have been made in biology in terms of being able to do single cell transcriptomics and, knowing every gene that's expressed essentially and every protein that's made allows us to really understand what's going on. So if we can get biopsies after treatment, we can begin to unravel all this stuff. And so that's what we're doing now is we try to work stuff out in mice, get a good idea it works, and then as soon as we can, with the help of Pam Sharma, who runs our clinical thing, go into patients, do a 12-patient trial. Safety and mechanism, not necessarily looking for an anti-tumor effect yet, but just did we at least not hurt the patient, and did we learn something about the mechanism? Maybe VISTA is the name of another one of these checkpoints. Maybe VISTA plays a role. Maybe the next time we need to add VISTA to the cocktail. So we go back and we add VISTA then. See if that works, And so that's the idea, is do this iterative thing. That's how we get it from 55% to 100%.
And my impression is, or my guess would be that this kind of therapy might also have the benefit that can, thinking of vaccines as an analogy, it sticks around in the body and
Once
might
you got
help
the T
prevent
cell,
what's coming next.
yeah, I mean, I think in a way, we do ourselves a bit of disservice by calling like the antibody that we made to CTLA-4. That's the drug. It's not the drug. It's the thing it's the prodrug. It's the T cell that's the drug.
Right.
And so that antibody's gone in a few weeks. The T cell's there for the rest of your life.
Okay, so we're running to the end of the podcast, but so I'll ask one
Okay.
slightly crazier question. I mean, all these ideas about networks and switches and nonlinearities. I'm a complexity scientist, among other things. It just makes me think of the study of complex systems. But is your work and the work of other people trying to do what you do, or is it just so focused on cancer and immunology that you don't have time to
No, I think no. That's an excellent question. But I think that if we can learn how to reverse this, we could treat autoimmunity. There's increasing evidence that a lot of neurodegenerative diseases involve dysfunctions in the immune system. If we can really understand what we're doing, we might be able to do something about Alzheimer's down the road, or Parkinson's, or diseases like that. I mean, we're thinking about that all the time, and hoping that some of the data that we have will just people. I mean, it's the very simplest oversimplifications. If we just do the opposite of what we're doing now, maybe we can cure those diseases. So that our institute our goal is to use the advances in basic science to advance medical practice. Right now, it's cancer. After we cure cancer, then we'll go after neurological diseases.
Never satisfied. Well, one of the goals of the podcast is to give young, curious people food for thought about areas that are exciting and changing very rapidly right now. You've certainly done that for us.
I think, of course, I'm biased, but I think that it's a wonderful time to be doing this work. And it's become a systems biology problem more and more. And so single cell analytical techniques, the ability to take a slide, now to diagnose a cancer, you get a piece of the cancer and you stain it with hematoxin and eosin, you get this purplish thing that you can look at shapes, and the pathologist could say, that's a cancer, that's not, there's some immune cells over there. Now we can look at that, and by doing some different analytical techniques, we can tell you every gene that's expressed in that cell. And what we already know is that the same cell that's here next to the cancer cell is going to be different than an otherwise pretty much identical cell over here that's not in contact with the cancer cell. And the immune cell, the same thing. The immune cell that's next to the tumor cell is not going to be like the immune cell that's in the blood. And so now we can start to unravel what are those differences, and how can we send the cells down there? We know that there are certain inhibitory molecules that the myeloid cells, particularly those that are in these complexes near the tumor cells, express to turn the T cells off. If we can figure out how to just turn those molecules off in the myeloid cells, then, but we got to know what they are first. I bring that up because I was just discussing with one of my postdocs 2 molecules of that sort that we recently found that we're in the process of trying to do that with. But as the science, as the engineers and the computational people and all that are coming up with all these magical new things that I couldn't even dream of 5 years ago. I mean, that's how fast it moves. There's stuff we're doing now that I wouldn't even have guessed that we'd be doing 5 years ago. That it's just such a fascinating time to be in biology. And I don't know, my motto is, I've always been fascinated in science and biology and everything. You might as well do something on an area of something that helps people. Because, I got into it initially because I just, my family situation. But on the other hand, it's fun, and it's rewarding, and it helps people too. And so I think that it's a lot of it's drudgery too, but it's just puzzle solving, and with the tools that we're getting that are coming, from bioengineers and computational people are just allowing us to ask questions that we couldn't even have dreamed of.
It's exciting times, absolutely. Jim Allison, thanks so much for being on the Mindscape podcast.
Thank you. By the way, I see you have a guitar. Do you play much, or do you?
That's a bass guitar that I'm very, very, very bad at. If we had more time, or if you want to take another 5 minutes, I was going to say, like, tell us the Willie Nelson story. Come on, it's so good.
Oh, I'll tell you one funny one. I had a knee replacement last. Was it last year? Yeah. Anyway, and Willie, I play with him occasionally, but he they asked me to come to Austin. I'm in Houston, his 4th of July picnics in Austin. So they said, "Would you come down?" I mean, he didn't, but his wife and his harmonica player is a friend of mine. So Jim, come down and join us. And I said, "I can't. I just had knee surgery, and I don't want to go down there." And they said, "Well, we just happened that they were playing at this place called the Woodlands, which is just north of Houston, near this lake where I have a lake house. And I was recuperating from the surgery there. They said, well, hell, we're going to be there on Saturday night. So why don't you come and play there? I said, well, I can't walk very well. They said, well, get a wheelchair.
Yeah.
You don't have to travel. So I said, okay. So my son got a wheelchair. We got some friends. We went down, wheeled me in. I'm still on the edge of the stage. And so I was waiting to play, but I still wasn't sure I could go out there. And one song came by, and they said, "Come out." And I said, "No, I can't. I can't stand yet." I was in a wheelchair. I had a cane. Anyway, Willie got into the gospel. He always closes his concerts with a gospel medley. "I Saw the Light," "Will the Circle Be Unbroken," "I'll Fly Away," all the great old songs. Anyway, so that's so much fun to play. And he said, Mickey, waves me out there. He had a microphone for me. I said, no. And Willie's wife got by. He said, "Allison, get your ass out." And so anyway, so I jumped up and took a couple of steps before I didn't even think. I just stood up and I had a cane in one hand, a harmonica in the other. And then I realized this isn't going to do because I need both hands when I get out there. So I turned and I threw my cane behind me and then turned back around and took a couple of steps towards the mic, and this woman screamed, "He's healed! Willie healed him! Praise the Lord!"
Evidence. It was not a double-blind study, but there's some data there.
And then Willie starts saying, "I saw the light." It was just so perfect.
Congratulations on being the recipient of a miracle. It sounds like you deserved it there. All right. Once again, Jim Allison, thanks very much for being on the Mindscape podcast.
Okay. Bye-bye. Thanks.